Unless you have an identical twin, no one in the world has the exact same DNA as you. This principle is the reason why DNA collection and analysis can be incredibly useful in criminal investigations. At the scene of a crime, the perpetrator will often inadvertently leave behind some biological material, such as blood, hair, skin cells, semen, and saliva, all of which contain DNA that can be used to try to identify the perpetrator. Amazingly, as little as 1/10,000,000th of a gram of DNA, or about 16 cells worth, is sufficient for forensic analysis. At a crime scene, biological material containing DNA is usually retrieved from objects using either cotton swabs or adhesive tape. DNA profiling involves the identification of unique genetic markers to characterize an individual's genome. Autosomal STRs, or short tandem repeats, are regions in the genome consisting of 3, 4, or 5 base pair sequences that repeat. The number of tandem repeats constitutes the genotype at STR loci. STRs are highly variable between individuals, making them effective tools for differentiating people genetically. The first step of DNA profiling involves amplification using PCR, or polymerase chain reaction. PCR heats up and denatures DNA, splitting it into 2 single-stranded molecules. PCR primers complementary to highly conserved regions on either side of STR loci binds these flank sites, and Taq polymerase uses these primers as launch pads to generate more strands of DNA. Replicating this process for several cycles allows for rapid and exponential amplification of STR loci. Gel electrophoresis separates samples by size, indirectly measuring the number of tandem repeats. Forensic analysts use STR genotypes to create a genetic profile for the DNA sample of interest.
There are 2 main ways these results can be used to solve crimes such as sexual assault, murder, and burglary. The results of STR analysis for the suspects and crime scene samples can be compared in order to find matches. This could be useful in identifying or ruling out suspects. In addition, STR profiles from crime scenes can be compared against databases of previous samples. These databases, which contain samples from both convicted criminals and volunteers, can be invaluable for finding suspects for crimes. It's important to remember that DNA evidence does not mean guilt or innocence. However, it can be used to show that an individual was present at a crime scene, or previously had contact with objects at the crime scene. Prevalence of DNA technology in media has led to what's known as the CSI effect, where members of juries place too much weight in DNA evidence. It's important to use other forms of evidence to provide a good criminal case, and for courts to inform juries that DNA evidence does not necessarily prove guilt. The way in which DNA can be collected and analysed for forensic purposes is incredible, and as long as DNA evidence is used and interpreted responsibly, it can be an extremely valuable tool in the pursuit of justice.